So we've got lots of definitions in these slides. Anything in purple or bolded is probably a really good definition that you want to remember. So do keep them in mind. Make flashcards out of them, I recommend. So I use Anki in uni, but in high school I'd use like Quizlet, or I'd like make physical flashcards. So you can make like actual flashcards and print them out and use them, and that might be a really useful way of studying. So what is gene expression? A gene is a particular sequence of DNA bases which encodes for a specific polypeptide chain. So these are inherited from a parent to an offspring, and they're kept within particular loci or locuses. Loci is the actual plural form of that word. And so what a locus is a fixed position in the chromosome where a specific gene is located. What is gene expression itself? So this is the transcription and translation of a gene. And so this is the process by which genetic material such as DNA is converted into a functional 3D protein. So it goes from DNA to RNA or mRNA, and this mRNA will then be translated into a polypeptide, hopefully a functional protein. So there are many different stages of gene expression. The first one is transcription, which occurs in the nucleus. So you have a pre-mRNA strand being formed from DNA. In stage 2, the pre-mRNA must undergo post-transcriptional modifications in the nucleus in order to become mature mRNA. The third step entails the mRNA moving to a ribosome. This can either be a free ribosome in the cytosol, or it can be attached to the rough ER. After this, mRNA will be translated at the ribosome into a polypeptide chain, and then finally that polypeptide chain will be modified, transported, and secreted or used within the cell. Cool, so this process here is just showing transcription. So you can see the DNA here is double-stranded. You've got the 5' and 3' end, you've got the coding strand and the template strand, and you've got the RNA polymerase, which kind of opens up. So it unwinds that DNA and opens it up to access it, and this allows the making of an mRNA strand, and you can see that's being made here. You can see that in DNA we've got 4 different nucleotide bases, A, G, C, and T, and in RNA we've got A, G, C, and U. And so these 4 bases are going to be used to create the messenger RNA.